All right, so thank you, Christina. So my name is Danny. I'm one of the research computing facilitators here at the HTC Path project. I'm going to be giving you a little bit of a story of our journey to supporting one of these AI/ML workflows, particularly working with AlphaFold over the past few months. And so typically whenever we're talking about AI workflows, the first thing that comes up to mind is GPUs. How do we get the nice H200 to run this workflow? How do I provision that GPU for the researcher? But oftentimes a lot of the pre-step, the pre-work for getting that work to the machine, the execution point, is a little bit ignored. And that's what a lot of our focus has been on for the past few months. And so I put getting a GPU is easy in quotations. Try and get an H200 today, you're going to have a hard time. But focusing in for this presentation on all the other stuff that we have to do in order to get that job to successfully run on a high throughput system. And so throughout the talk, we'll be going over how we're using this concept of translational computer science and having that constant feedback with our community of users that feeds back into how we change our practices, how we change our infrastructure, how we change our software here at CHTC in the Path facilities. And so a little bit of context for folks that aren't familiar with AlphaFold. So AlphaFold is a software that came out, AlphaFold3 came out about 2 years ago. A couple versions have been going back since about 2017, 16. A couple years ago, it won the Nobel Prize in Chemistry. And it's a software that's been able to resolve a lot of the protein structures or the biomolecule structures across life. And the latest version of it is AlphaFold3, which can now do proteins, DNA, RNA, all the nice biomolecules that we see, as well as looking at interactions between these biomolecules. And so under the hood, AlphaFold is sort of a two-step process. We have a data-intensive pipeline where AlphaFold skims through these massive 750 gigabyte data sets of biological sequences, and it produces these things called alignments. And so these are sort of your control find of biology. And so in order to complete this process, you need to have these data sets, these databases of about 750 gigs, can take several hours at times, depending on how deep your alignments go. It's really just CPU going at it. It's very limited on how many numbers of CPUs it could use. And oftentimes we saw users just submitting this to a machine, throwing an A100 and 8 cores at it, and it would spend a few hours in that CPU period not actually using the GPU, and we just had that GPU idling around. After that CPU step, once you've formed your alignments, you then have that serve as the input to a neural network, and that is going to actually produce the predicted protein structure using model inference. This step doesn't require the databases at all. It's a very short runtime unless you're doing something wacky, which we'll talk a little bit more about later. And generally, you're looking at a runtime of about 20 to 30 minutes. And so once you run this workflow, you actually get a protein structure that a lot of biologists, biochemists can then start to do their additional analysis afterwards. Now, a couple years back, AlphaFold started to come into the fold with CHTC here at UW. We had a researcher, Samir, who worked with the GOVRC, and he had developed this repository that allowed AlphaFold to run on CHTC infrastructure. And while it worked, there were definitely some areas where we could do a little bit of improvement. One of the big things that it did that made it very difficult to scale up was it actually shipped those databases back and forth for every single job. Beyond that, there is these high disk allocations that we had to set up for each job. So if you send out 10 jobs, you had to find about 10 terabytes of disks that you could pre-allocate for these jobs. So it became very cumbersome to transfer this data and get it to the EP when it was needed. And so about a year ago, Raison from the Mortgage Research Institute came down from the Singh Grant Lab and said, hey, I have this research project I'm trying to get to. We're looking at the protein structure for a multi-chain conformation for a viral protein. It is about 1,000 amino acids each chain. And I want to look at the 11, 12, and 24-mer version of this structure. So that's 11, 12, and 24 copies of this chain in a single structure. And as soon as Raison said that to me, I was like, ooh, that's a massive structure that we're going to be trying to predict. Very discouraged in general when I heard that at first, but I was like, let's see what we can do. And so we started to look at AlphaFold, started to take in that repo and dissecting what was happening under the hood with AlphaFold. We realized there was a lot of limits to where we were able to support it. Its current instance, at the time, this was a repository that was managed by someone else, and we didn't have a lot of visibility into it. There wasn't a lot of institutional knowledge about how AlphaFold worked. We didn't understand GPU memory, runtime, the job shape in general. And so our support confidence wasn't that great. And so when we start actually looking under the hood, that's when we start to untangle some of the areas for improvements that we wanted to make. There's a lot of fuzziness around how a job shape is defined, how big of a GPU you need, how many cores you require, how much memory you need for that first part, how we would handle the databases. There's a lot of documentation gap internally for facilitators, and there was this constant issue of we didn't have ownership of the repository. And so say about 7, 8 months ago, we decided, all right, we're gonna actually start supporting this a little bit more in depth. And we took over the repository and started working on supporting it a little bit more first class. And so the question started to shift from, can folks run this? We had seen that folks were running this. We had different researchers across CHTC that were running this at all times. And the question moved towards, how do we better support this? How do we support this in a way that's in line with sort of the principles of high throughput computing and scaling your workflows up? The first thing that we had to do in order to achieve this was understand that job shape. How do we advise researchers on how many cores they need, what GPU they need, how much memory, disk, the runtime. If we have a user that is submitting a job that's running for 20, 30 minutes, I might sit there and say, hey, go ahead and throw backfill on there. There's a possibility you're going to get interrupted, but 20, 30 minutes is a short enough time period that you won't waste too much time. Whereas if you have a job that's running for several days, might be a little bit more discouraged to use backfill. And so we started to benchmark AlphaFold, understand what the shape of these jobs started to look like. And one of the areas that we started focusing on, particularly with Raison's work in mind, was understanding the requirements for GPU memory. And so there's a basic sort of hand-wavy idea online of you can run about 5,000 tokens on a A100. That gets you about 5,000 amino acids. So we could have a rough idea of what that looks like for a user, but we wanted to have sort of a back of the napkin better metric of, if you have these many amino acids, you might be better suited with targeting some of our RTX GPUs that we have a much higher availability of them, instead of trying to compete against everyone in the world for an A100, H200. And so we took a monomer of the human actin protein and started adding copies of actin. So it's about a 300 amino acid length protein sequence, and it forms really long fibers. And so each time that we added one more copy, we looked at how much memory it was trying to use during the run. And throughout this process, we were able to establish with simple linear regression an idea of, all right, if you have 5,000 amino acids, you're going to need at least this much GPU memory. This was able to help us understand what types of GPUs could support what workflows or what size of workflow. And one of the things that we realize is a lot of our common single or double or two protein interaction runs could actually fit on much smaller GPUs. They could fit on an ICV100, they could run on an RTX GPU that we have pretty good capacity of and tends to be fairly idle. Whereas when you need those massive 11 mer-ish structures, you might be targeting something like an A100 or an H200. And also helped us start avoiding some of these out-of-memory failures. We were able to start advising researchers and say, hey, if you're gonna submit this massive structure, don't try and target a V100. You're gonna have a bad time. And it gave us a better idea of what we could tell folks when they would come to us with the proverbial question of, how long is this gonna take to run? And a lot of times we're not sure. Depends on the capacity of the pool, it depends on who's using it. But we do have an idea of our H200s, you might be looking at a couple days. Our A100s, you might be looking at a day. RTXs, you might be looking at a couple hours to minutes. And so we were able to start getting folks some more information. So I went back to Raison and said, all right, so we ran the numbers and it looks like for your 11, 12, and 24 mer, you're gonna need 190, 210, and 425 gigabytes of VRAM. Fortunately, AlphaFold does not run on multiple GPUs, which means you need to find a GPU that has 425 gigabytes of RAM. At the time, and I still think today, that does not exist. And so the question then became, can we start using things like unified virtual memory? And so that started a second sort of iteration where we started looking at how do we keep pushing past our GPU capacity. And going through the documentation for AlphaFold, if you've ever had to subject yourself to that, I'm so sorry. There is a lot of inconsistencies, but one of the things that we found was JAX, which is a library that runs under the hood, is able to use unified memory, allowing the job to spill over into the host RAM. And we started to troubleshoot this, and we realized we were actually able to start supporting these unified memory workflows. But the one caveat that when you have a multi-tenant system, particularly with JAX and the way it sets up, it needs a contiguous memory block. It's really hard when you have 10 other concurrent jobs running on that machine that's fragmenting the host memory. And while you might have a terabyte available of memory on that machine, it's fragmented all over the place, which made it very difficult to start these jobs. So we moved towards a different approach for starting up these workflows where we would have the machine get drained. So we would stop sending jobs to that GPU node. We would reprioritize that machine for that user, allow the user to match, the job would start up, and then any other jobs that were in the pool could backfill on the machine once that memory was already reallocated. And I'd say after about 3-4 months, we finally had victory. We had a structure. This was a wild journey to get here. But we were able to start predicting or producing the AlphaFold structures for Raison. And so we were able to take a workflow that was previously impossible, that we were just like, yeah, we can't support this, and started moving it towards this area where we're able to support it with a lot of tender love and care. There was a lot of RCF involvement. There was a lot of involvement with Brian Lynn's folks, Moat in particular, in getting these workflows up and reprioritizing the machines. But this wasn't where we want to stay for these workflows. Eventually, we want to move folks to being able to be self-sufficient and be able to run without having to have that direct intervention with an RCF. And so this started to open up additional questions of how do we continue to support this at scale? Along this entire path, we had the long ongoing issue of the data problem. We have still these 750 gigabytes that we have to move back and forth. And with our current structure, for every single AlphaFold job, we would have to move a 750 gig database to that EP for every job, for every user, for every site. So this pretty much made it impossible to run on the OS pool. You try and find a slot on the OS pool that has a terabyte of disk, you're going to have a handful at most. And so it really limited on the OS pool, and even on CHTC, this was not scalable. So we started to ask ourselves, can we start to think of data sets as a property of a machine and as a property of infrastructure? We started to explore this idea of using data set as a matchable resource, something that Condor knows, hey, this machine has this data set. I should go ahead and send jobs that need that data set to that machine. And so the idea was user would request a data set by saying, hey, I want to match the machine that has the AlphaFold data set. It would be sent over to the matchmaker, and it would be sent over to AlphaFold machines that have the AlphaFold data set. And so we started to think about this. We started to notice that this is an ongoing problem, not just with AlphaFold, but many other workflows. Originally, I'm a biologist. One of the big ones that we always use is BLAST. BLAST is also insanely large in terms of its databases. And so this was one of those generalizable contexts or generalizable practices that we could start to apply to other workflows. And so the idea was if you had an AlphaFold job, send it to the scheduler. Scheduler would look at it and say, in my pool of resources, I have machines that have AlphaFold. I'll send it over there. Let's say you're running Cocoa, object classifier, and you need the Cocoa data set. You'll send it over to the machines that have Cocoa. Now you have a bunch of jobs from a researcher that says, hey, I don't need any data set. I'm cool to run anywhere. Send it to the scheduler, and the scheduler will send it to any of these machines. And so still that person that's running a sort of data agnostic workflow is able to match at a higher rate. But we're also still able to support these more data intensive workflows. And so we started piloting this on the OS pool, and we started here locally first with CHTC, but also working with our Jetstream 2 contribution. And we were able to place the data sets on a manila folder on the Jetstream 2 allocation and start serving it on the OS pool using our matchable resources. And so we're looking to chat with other campuses. If you're here this week and want to discuss sharing some of these data sets, if you happen to have the AlphaFold data set, come talk to me or one of the other facilitators. But we'd love to chat a little bit more about what this could look like in other sites as we start to scale out. And so at this point, we had started to move our sort of envelope of support over and over. We now had a better idea of supporting sort of the standard single, double, duplex protein structures, fairly easy. Once we start to get into larger structures, we might need a little bit more involvement. When we got into these massive structures like Raison, we had an idea of how we could support it. It was still very involved with us. And so we wanted to go back to the community and say, hey, we've been doing all of this work for the past few months. Let me hear from you. We want to know what you guys are interested in. And we want to share the insights that we've gleaned from this process. And so back in February, we had a training here at CHTC for running AlphaFold on CHTC, and we had over 75 attendees. We had folks standing. Sorry about that. We had folks that were standing in the back of the wall, folks sitting on the floor. It was very clear that people want to run these workflows in our local community. And even throughout our interactions with folks on the OS pool, I would say at least a couple times a month, we get an email of someone that's wanting to run this workflow. And one of the things in our interactions with researchers, I don't know why the text is messed up. But one of the things that we realized with researchers as we were starting to chat with them is while we had done a lot of work with Raison to support this sort of one-off, bespoke, ultra-large structure prediction, people wanted to submit high throughput screenings with AlphaFold. They wanted to run complexes across hundreds of thousands of proteins and use it as almost a process akin to docking, for those of you that might be familiar with that workflow. And so, again, this started to shift the question of how do we run these massive workflows? How do we support AlphaFold at CHTC and the OSPL? To how do we start to scale this out even further? And so there's a researcher here at UW-Madison, Kavi Mehta, who came to us and said, all right, I study human-virus interactions, and I'm interested in complexing the full human proteome. It's about 20,000 proteins with the full proteome of HPV. It's about 9 proteins. That runs you at about 180,000 pairwise predictions. I was like, that's fun. That sounds like real high throughput. Let's go ahead and see what we can do in this area. And so started chatting with Marone, with some of the other folks at CHTC. And again, we started to move from these one-off predictions where we would spend so much time computing these alignments to how do we start to reuse the components of these alignments that we're spending so much energy in trying to find the machine that can run it with the database that has the resources. And so I started doing a little bit more research, and one of the things that we realized is those alignments that come out of the first stage can be reused. They're on a per query basis. If you have 2 queries in your sequence, you're going to produce 2 alignments. And so you can reuse those alignments across different workflows. And so conversations with Marone, we had the idea of what if we set up a sort of library of alignments that CHTC and Path staff are contributing to and building for other folks to start using. And so the idea was researchers would come in and submit a standard AlphaFold workflow. It would check the library and say, do I have an alignment for this already? If it does, it would serve it back to the researcher. The researcher could opt out or opt in at any point. And they would be able to reuse those alignments across their workflows. So going back to Kavi, we had sort of the standard n times m combinatoric prediction, where we had 20,000 human proteins and 9 viral proteins, crossing them together for 180,000 predictions. What that ended up actually resulting in is being able to reduce the number of alignment jobs that we had to run from 180,000 to 20,000. In terms of actual compute time, that was a reduction from about 720,000 CPU hours to just under 100,000. So it was an 89% reduction in that alignment step. And it was a faster way to get the researcher to that inference point, which is where their science actually was happening. More importantly, this is starting to open up doors to workflows that were previously impossible. So this is a researcher, Hanan Shaban from Florida National University. A little bit of context, Hanan was actually my former undergrad when I was a PhD student. And Hanan studies the molecular mechanisms of how algae, microscopic algae, become endosymbiotic with cnidarians. And cnidarians include things like jellyfish, corals, sea anemones, all the fun, weird creatures of the sea. And if you've ever studied anything with algae, plants, they have messed up genomes. So there's about 25,000 genes. That's a very common number, 20 to 25,000 in animals. Algae can go anywhere from 40,000 to, I've seen something as high as 100,000 genes. The specific model that she's working with is 25,000 genes in the anemone, about 40,000 genes in the algae. And that comes out to about 1 billion pairwise alignments. With this library, we're able to reduce that to 65,000, which takes a job that was previously impossible. We would simply look at a researcher and say, this is not something that any cluster could support, and actually operationalize it on the OSPL. And so looking at the AlphaFold library cache and where it is today, so it's very early on in its beta testing phase. We're releasing it to very trusted users / victims that are interested in giving us that feedback. And we have about 400,000 alignments already cached in that database. That represents about 320 unique sequences. There's a little bit of overlap from alignments that are coming from different sources, like Google DeepMind, who developed AlphaFold, alignments that we are developing, so on and so forth. It's represented, or we have represented about 10,000 organisms, and we're actually now starting to see community contributed alignments that we're actually getting from researchers on CHTC and the OS pool. Further, we've seen that in about the past year, we've seen about 500,000 jobs on the OS pool and CHTC that have been running AlphaFold. And that's been across about 70 users. I would say, again, every month we see new users that are interested in running this every day. We've had this massive flagship training, and we're working towards growing this further, working with Tony Gitter's group and Hannah Wayman-Steele, who'll be talking later on this week about supporting researchers in understanding which models to choose and what the caveats between those are. And we have this pre-staging of data sets already live, at least on one site on the OS pool, as well as here at CHTC. Now, in order to support this, we've had to re-envision a lot of the work that we've been doing. So this has required a lot of workflow-aware facilitation. It's been getting in the weeds with a researcher and saying, all right, how are we going to support this? It's included working with infrastructure, working with the folks on the HTCSS team to re-envision what we can do with data. It's been talking with the Pelican team, who backs the alignment cache, and how do we serve this data correctly to the user, and preparing that meaningful documentation to researchers. And so overall, we've been able to take this translational computer science approach and have a researcher here say, hey, I want to run hundreds of thousands of jobs. And with our own background as researchers or research computing facilitators, understand what those limiting factors are, having the discussion with the team and saying, all right, we need a way to support reuse of artifacts, creating those changes on the infrastructure, and being able to help support researchers running their workflows. So all in all, in order to support these sort of next-gen AI, ML-enabled workflows, we are going to require this approach of, or we need this approach of translational computer science, where there is that constant communication and, more importantly, innovation between researchers, facilitation, software, data, and infrastructure professionals across research computing. With that, I'll take any questions that you might have. Any questions? Steve.
So you gave a hint on your last slide. You were using Pelican to cache that 750 gigabyte database. But I have a couple follow-ups from that. When you say that Coco or that the AlphaFold3 is available on a node, was it available just in the cluster and shared between nodes, or is it all fetching it from Pelican, or was it on every node?
Yep. So the data set is not actually, the databases aren't served by Pelican. What's served is the alignments for the library of caches. The way that we're serving data for these large data sets is saying, hey, these nodes already have it, whether they're mounted onto the node as a network file system. However, different sites are managing their own storage and allowing sites to let us know, hey, I have this data available on this machine.
Right. So if it's a database, yep, is every job reading all 750 gigabytes of that? Or?
Yeah. So AlphaFold reads pretty much head to toe for the databases, with the exception of a handful of files.
And the site admins don't shoot you for doing that much disk I/O?
I'm sorry?
The site admins don't shoot you for doing that much disk I/O? I would.
You know, Brian Lynn has chased me down a couple times, but that is part of the feedback that we're getting. Right. Of how do we apply concurrency limits so that we don't wreck a network.
And then the follow-up is, you had 75 eager participants there. As all of them have their 750 gigabyte database, or in physics, your 10 terabyte database, yep, or this special thing or whatever, how do you organize that in some reasonable way
Mm-hmm.
so that you don't totally, I mean,
Yeah.
these are things that Dune is hitting with Stash Cache K Pelican on a regular basis on bigger scales already. So I'm interested to hear your input.
Yeah, so at the time, and this is part of also a little bit of the impetus, currently I know a lot of the practice with our folks at CHTC is you have a copy of databases, things like BLAS. We might have hundreds of researchers. Everyone has a copy of BLAS. They never update it. And you end up having a lot of this data redundancy. So this was an approach for us saying, all right, we're going to maintain a copy of this data set in order to reduce our footprint on our storage, but also allow folks to access it in their jobs directly.
So before we go to lunch, if you have on your campuses researchers that have interest in certain proteins, please let Danny know so we can direct our library generation to these proteins. So anyone who has a shopping list of proteins, which are very popular these days, then please let us know and we'll try to direct the efforts towards these proteins.
I think especially with what one of our previous speakers said, you know, as RCD professionals, we are sort of the communication to our researchers of, hey, there's this resource that exists. Hey, the OS pool exists. Hey, the OS pool is supporting AlphaFold. If you have researchers that are interested, please share that information with them. They can contact us. We'll work with them. We want to get in the trenches and support them.
Yeah, so maybe I didn't understand exactly that you did a lot of trial and error in this GPU benchmarking to do that. Can you like describe this like procedure? Because this is where let's say I have some problem, I get stuck. So what does this benchmarking mean and how do you like then go from one, like how do you scale? Like, okay, I guess you do like small steps and then you scale it and does it work all the time?
Yeah, I mean, the way that we established that range was we took a protein that was very well characterized. We had an idea of it's small enough that those steps were not leaps. So we had enough fine grain. And with AlphaFold, it's great. There's a JSON. You add yet another letter in the chain ID field, and it'll add another copy of that sequence as it's starting to predict the structure. So what we ended up doing was submitting hundreds of jobs that were going from a single copy of actin, that protein, to I think the highest that we went to was about 25, 26 of them. That's when we started to hit the limits of like our H100s, our H200s hadn't come up. And so we use that to measure the memory that was being used in the job and have an idea of, all right, if you have these many tokens, you should expect this much memory usage. Yeah. Yes, so that's all coming out in the log file. There was a little bit of fiddling that we had to do with like NVIDIA SMI to get like nicer data as the job was actually running. But we were able to just get it at the end of our standard Condor log, where it would say, this is the memory that you requested, this is how much you used.